everyone, and welcome to today's webinar, Development of a Novel Targeted NGS Panel for Analysis of DNA Methylation Biomarkers in Lung Cancer Research, presented by our speakers, Dr. Lucia Muscarella and Dr. Cristina Guillén. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, visit thermofisher.com/AmpliSeq. Now, we encourage you today to participate by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. But before we get to our speakers, I'd now like to welcome Dr. Giorgio Pea from Thermo Fisher Scientific to give an overview of the custom AmpliSeq solutions they offer and to introduce our first speaker. Welcome, Dr. Pea.
Hello, everyone, and thank you for attending this webinar. My name is Giorgio Pea, and I am the Global Product Manager for Custom Application in our Clinical Sequencing Division at Thermo Fisher Scientific. In this event, we will learn from our speakers about how a custom AmpliSeq solution was leveraged for an advanced research application study on DNA methylation biomarkers, and then about how our analytical validation team can help to accelerate the adoption of AmpliSeq-based workflow in clinical research laboratories. Before leaving the panel to our speakers, I wanted to briefly provide an overview of the multiple applications that custom AmpliSeq solution can cover for your research needs through flexible targeted NGS workflows. As schematically shown here, custom AmpliSeq applications span across several areas, going from cancer research, including gene fusion detection, to inherited disease and reproductive health research, and then again from gene expression analysis to microbiology and infectious disease research. And to targeted DNA methylation analysis, as our next speaker will so nicely illustrate in our first talk today. Ion AmpliSeq technology, developed and constantly innovated at Thermo Fisher Scientific, is globally recognized as the gold standard method for amplicon-based targeted NGS testing. Its key features, briefly outlined in this slide, include but are not limited to small input requirement, scalability, and versatility, plus ultra-high sensitivity with the AmpliSeq HD chemistry. And further combined with our full customization capabilities, either through AmpliSeq.com or our manually curated white glove custom design service, the Ion AmpliSeq technologies allows addressing many advanced research needs using targeted NGS assays. A nice example of this is the ability to interrogate with great details and accuracy the DNA methylation status of multiple targeted CpG positions in DNA samples, and in doing so, establish their potential utility as biomarker for several applications. So, without further ado, it is my pleasure to introduce you to our first speaker, Dottoressa Lucia Muscarella from Casa Sollievo della Sofferenza Foundation, Italy, who will present her experience and work with a custom AmpliSeq methylation panel for the analysis of DNA methylation biomarkers in lung cancer research.
Thanks to Giorgio for its introduction, and thanks to Thermo Fisher. And thanks to all the organizer and provider for making this possible. It's my pleasure to be presenting you today on the topic development of a novel targeted NGS panel for analysis of DNA methylation biomarker in lung cancer research. My name is Lucia Muscarella, and I am a group leader at the Laboratory of Oncology of Casa Sollievo della Sofferenza Foundation in Italy. Before we get started, this is a disclaimer by Thermo Fisher Scientific, the sponsor of this webinar. Today, we will talk about the epigenetic modifications and their role in human diseases, how we can measure epigenetic variations, and the use of next-generation bisulfite sequencing for DNA methylation analysis. Finally, I will show you the design and testing of a novel Ion AmpliSeq custom panel for methylation study. We can start by saying that the concept of epigenetic has changed several times, and the actual definition is that an epigenetic trait is a stable, heritable phenotype resulting from changes in a chromosome without alteration in the DNA sequencing. In mammals and humans, there are many different epigenetic modifications, ranging from DNA methylation, histone modification, and small non-coding RNAs. Among these events, DNA methylation is one of the most early epigenetic modifications. Two of DNA of 4 bases, cytosine and adenine, can be methylated by specific enzymatic activity at frequently CpG dinucleotides. CpG sites are not randomly distributed into the genome in mammals and in humans in general. CpG-rich regions, also called CpG islands, tends to be frequently methylated and tends to be located at specific gene regions, such as transcription starting sites, as promoter region within gene bodies, also called intragenic, and between annotated genes, also called intergenic CpG islands. Variation in DNA methylation at CpG islands can alter gene expression, thus leading to a decreased transcription. Epigenetic was firstly recognized in relation to tissue development and cellular metabolism, but it also been related to several key biological processes, such as X chromosome inactivation, genomic imprinting, and aging. From a pathological point of view, epigenetic variation may play a leading role in autoimmune disease, neurological disorders, metabolic disorders, and also in cancer. Actually, the DNA methylation is one of the most recognized epigenetic markers that led to a massive distortion in cancer cells. It has been observed that a large number of aberrant methylation events occur simultaneously in a group of genes, thus providing a growth advantage to the cell in promoting cell differentiation, heterogeneity, and neoplastic transformation by inducing angiogenesis, activating invasion and metastasis, changing energy metabolism, and escape immune and apoptotic system. Due to this reason, methylation profile has been suggested as a promising cancer biomarker. By consequence, one of the main discussed questions in this field is actually how to study DNA methylation changes and how to find the optimal approach to use, starting from different types of samples. We can use two different groups of technical approaches, that is genome-wide approaches and targeted approaches to study specific candidate regions. Genome-wide approaches can be used to study primary tissue biopsy samples or blood samples, whereas targeted approaches can be used to study many types of samples, such as samples obtained from non-invasive techniques and from invasive protocols, such as fluid or tissue biopsy. Among these targeted approaches, the targeted NGS can be considered one of the most comprehensive methods to analyze candidate gene for multiple regions and for large screening study. With an increasing number of clinical actionable targets, NGS and the optimization of next-generation sequence technique enables a high sensitivity, specificity, and time saving of analysis of nucleotide changes in multiple target gene regions at single base resolution. Starting, moreover, for a relatively low quantity of biological materials, the NGS approach offers in the field of epigenetic the great advantage to successfully quantify DNA methylation density at single CpG site at any genomic location. Now I present you the design and the first step of validation of a novel ion-ampli-seq Opera METI-PAS custom panel for DNA methylation analysis. The Opera METI panel was designed in collaboration with the White Glove Team Service of Thermo Fisher Scientific and was designed to simultaneously analyze at single base resolution the methylation level of many CpG sites of cancer-related gene. The Opera METI panel include genic region annotated as having show value in peer-reviewed scientific research papers. More specifically, the panel includes 155 genome regions, 1107 CpGs located in 80 cancer-related genes, linked to immunotherapy, oxidative stress, and prognosis in lung cancer and in other solid tumors. Amplicons have a size of about 100 base pair, and amplicons were designed for both Watson and Crick strands. For each gene included in the Opera METI panel, both island located and interspersed CpGs at both promoter and gene body region were targeted. And this is why gene body methylation is widely involved in the expression regulation of many genes and is becoming established as an important biomarker in clinical cancer research application. To experimentally validate the performance of our NGS methylation panel, we decided to test 3 different types of biological samples having different grade of fragmentation. Specifically, we used FFPA paired tumor non-neoplastic lung cancer tissue, 3 paired tumor non-neoplastic OCT embedded tissue, and 2 cell lines from the ATCC collection. Bisulfite conversion of gDNA was employed as a second step of our analysis in order to discriminate methylated versus unmethylated cytosines. The performance panel indiscriminate different rates of CpG methylation was assessed by running commercially available fully methylated and fully unmethylated gDNA and mixtures of them. Before starting the bisulfite conversion, unmethylated lambda DNA was added to each gDNA sample in order to discriminate bisulfite conversion efficiency during samples treatment. Library preparation and sequencing was made using the standard ion-ampli-seq amplification protocols, starting for different quantity of bisulfite converted gDNA. Library was then quantified and qualified using real-time PCR, qubit, and tape station. And finally, library were loaded on Ion Chef instrument, and sequencing was performed on Ion G Studio S5 system workflow. This is the result from pre-sequencing quality control check analysis. And the Opera METI methylation assay allowed to obtain libraries starting from gDNA extracted from different matrices and bisulfite converted. No substantial differences in the library quality and length were observed among converted gDNA template from FFPA and OCT samples. In terms of concentration and size distribution of amplicons, a higher library quality was obtained using a minimum of 10 nanogram of converted gDNA. Since lower gDNA input does not ensure good performance of library synthesis. The empirical calculation of a cutoff of minimum required mapped read per target was made according to the following criteria. More than 20 methylated read for testing CpG sites, more than 40% of tumor cell content, and more than 10% methylation level per site, and was also related to an expected conversion efficiency greater than 99%. We can therefore assume a minimum of 500 total mapped breeds per site to achieve a successful run site. Based on the experimental sequencing performance of sample, we assume that a minimum of 6 samples can be loaded in the same run. For our tested sample, we obtain a very promising and comparable results to those already published for methylation analysis by NGS approach using the targeted AmpliSeq technology in terms of mean depth, average number of mapped breeds, and concordant results between expected and observed % of global methylation for all targeted CPGs. The methylation analysis torrent suite plugin was used to analyze the sequencing output of Opera Meti panel and annotated percentage of methylation of each targeted CPG site. The methylation plugin allows sequencing breed alignment onto the Watson and Crick strands using the reference genome. Status check CPG methylation was made in a strand-specific manner, and the reports and the text file were generated for each amplicon. The text file contained the number of methylated breeds, unmethylated breeds, and % methylation for each amplicon. Moreover, the summary report was created for each sample and include barcode name, the sample name, the total number of breeds that cover the targeted CPGs, and the percentage of methylated breeds. The methylation analysis plugin allows the evaluation of performance of biosulfite conversion rate and global methylation. A high conversion efficiency as a result was obtained for all treated control samples, with a mean of 98% of conversion efficiency for all samples. Methylation data obtained from the FFPA samples were used to evaluate the utility of the MET-E panel to visualize the CPG methylation distribution along different gene regions. And as expected, variation of methylation level at different CPG sites for many genes were observed between paired, normal, and tumor samples. Moreover, and finally, the Opera Meti panel allows us to visualize the asymmetric distribution of methylated CPG at Watson and Crick strands. Using the methylation analysis plugin, we can manage data obtained from the panel differentially, and differentially methylated cytosines can be found along the same gene and in each gene in both double-stranded and single-stranded context. The ability to determine the exact location and status of CPG methylation in Watson and Crick strands separately could provide interesting experimental evidence for innovative clinical cancer research application. CPG positions are in fact usually expected to be either fully methylated and fully unmethylated in both strands. Therefore, there should be a right or wrong strand when choosing to study conventional methylation patterns, and that obtained from one strand can be safely assumed to apply also to the second strand. Anyway, recent studies show that methylation in a CEG context is not always double-stranded. It can also occur on just a single strand as described in solid tumors, and should be related to gene function and transcription. Concluding, we can say that the epigenetics is relatively new compared to the genetics and is being evaluated for potential futuristic clinical research application and methods. Starting from a relatively low quantity and quality of biological materials, the NGS approach offers in the field of epigenetic the great advantage to successfully quantify DNA methylation density and its differences at single CPG sites of both promoter and intragenic regions that can be specifically customized based on their relevance on the research study. While the main focus of this study was to investigate the methylation status of multiple cancer-related genes by using a custom NGS panel, our analysis also offered new insight into the heterogeneity of CpG methylation patterns among genes and samples. The final translational advantage of this feasible high-throughput approach to study epigenetic fluctuation will be the opportunity to construct a more robust and integrated algorithm to predict the disease evolution in cancer patients. Let me thank all the staff of my foundation for their support in the study, Dr. Fabrizio, who led the experimental workflow, and Dr. Giorgio Pea from Thermo Fisher Scientific for his support in the design of Opera MetiPanel and the definition of analysis pipeline. Thank you.
Thank you, Dr. Muscarella. Our next speaker today is Dr. Cristina Guillén, Senior Technical Project Manager, Analytical Validation Service, EMEA, at Thermo Fisher Scientific. Dr. Guillén, you may now begin your presentation.
Thank you. I'm pleased to present you the Analytical Validation Consulting Services for Research Use Only NGS panels from Thermo Fisher. Analytical validation is defined as the use of an analytical technique, process, and procedure to generate results in a documentation form, assuring that all parts with a workflow are suitable for their use. If we continue with this definition, analytical validation is an industry-accepted process, tests a procedure or a workflow using analytical methods, documents the results with tangible evidence, proves that the workflow or procedure are suitable for its use. If you are asking yourself how often you should perform an analytical validation, the recommendation will be that you are doing so every time that you are adding a new test to the menu, every time that you have a major change in the workflow, you are adding new tissues or targets to the test, or obviously when you are adding instruments or updating instruments in your workflow. The Analytical Validation Consulting Services for Research Workflows from Thermo Fisher are helping customers to go through this process. We are supporting customers that are running NGS assays and want to validate them in an ion taurine NGS system. You can see here some of the examples of the panels that we are supporting for the analytical validation. In the area of somatic oncology research assays, we are supporting customers through the validation from panels like the Oncomine Focus assay, the Oncomine Myeloid assay, or the Oncomine Precision assay. In the area of inherited disorder and reproductive health research, we are supporting customers that are running validations on pharmacogenomics panel, ion amplicin carrier-seq, and of course on that they are running their validation in an ion amplicin or an ion amplicin HD custom panel. When a lab is going through an analytical validation, usually they will need to cover most of the steps that are here. That will start with the project planning. The lab is going to decide which one is the assay that they want to validate and to see if this assay is really fitting with the workflow that they have in their lab. In some cases, they will also not only start with a new assay, but also with an instrument. So they will need to do or take care of the installation and the qualification of this instrument. When this part is finished, is starting the real analytical validation with a pre-validation planning where the lab will define the analytical validation plan. They will the protocols to follow and will select the controls that they need to run through the validation and to test them before starting with the real experiments. Once the controls have been selected, the plan is done, the real validation and the real running of the samples will start in the lab. With the data collection, once this part is completed, all the runs are done, the lab will be involved in the analysis of the data, and preparing the final validation report. In some cases, after the validation is finished, there will be a need of additional tests, and if the lab has decided to do so, it will start the phase of the accreditation and reporting. How can Thermo Fisher help customers here? The analytical validation consulting service is helping customers through the process from the pre-validation planning to the finalizing the validation report. We are there to prepare the draft for the validation plan, they help customers with the protocol, select the controls that are the ones, the proper ones for the assay that the customer is running. We are supporting customers through the runs that are done in the lab, through the data collection, and finally we are analyzing the data and preparing the final report. We really help you to save time and to save cost. We are helping you to save time because we are helping you to launch the NGS assay as quick as possible, and that the lab run start running profitably. We are helping you to save costs because we have an effective validation strategy that you don't need to start doing a lot of repetitions to test the controls before you can start really the real validation. We are training your staff, and we are helping you to execute the plan. We are also keeping updated for the tester about the testing requirements that are needed in a lab, and knowing the documentation that a lab will need through the analytical validation. The critical personnel that is going to be involved in the project through an analytical validation will be, first of all, obviously the project manager that is responsible for the project, and to run as smooth as quickly as possible that the customer can launch the assay in their lab. We are supported by the bioinformatics team, which will be the ones that are helping us and helping the customer to understand the data and to understand the analysis that is being done. If the customer needs a little bit more of support from the bioinformatics part, we have also bioinformatic packages that a customer can select. The application scientists will be critical also for the analytical validation because he's really the expert in the workflow. They will be the ones that are responsible for the workflow training and the optimization on the assay at the customer side. Once the analytical validation starts, we will have what is called a customer kickoff call. In this call, we will discuss with the customer the analytical validation process, the timelines that are required, and review the analytical validation plan with the customer or with the lab. Once we have this first kickoff call, we are going ahead with what is called the lab readiness. Here we need to know that if there's a new instrument, the instrument has been installed, has been qualified, and also we need to make sure that we have the controls, the reagents, everything that we need to start really running in the lab. Once we know that the lab is ready, we will start with the training. The training is going to be performed and covered by the field support specialist. And usually what we do is that we start or we do the training at the same time that we do the first analytical validation run. That means that we are increasing the efficiency and we are reducing the cost for a customer because everything or both of the things are done at the same time. Once we have the first analytical validation run and it's completed and the data has been served, the project manager will analyze the data and we plan and is responsible for planning the next run. That will be always happening following weekly analytical validation calls in a way that every week a customer will have the new plan for the new run, until we are at the end of all the runs that should be done for the analytical validation. When this is completed, we will finalize the data analysis and we will prepare the analytical validation report. At the moment that this is done, we consider that the analytical validation is completed. We have three options for the analytical validation consulting service for research use only assays. The first one is called, or is the analytical performance verification. In this case, this verification is designed for customers that perhaps they have, or they would like to know the performance of an assay before starting a full validation of with this assay, or perhaps they have changed something in the workflow of their assay, and they want to do a verification of this change. In this case, in this package are included the controls, the workflow guidance, the data analysis, the consultation with an analytical validation specialist, and at the end, the customer receives a summary template. The second option is the analytical validation regional. This one is designed to support international testing and validation requirements. A project manager in this case is assigned to the validation, includes the workflow training, guidance and optimization, template documentation, controls, data analysis, and at the end, customers receive a validation summary template. The third option is the analytical validation, which is a full validation, same as for the second option, but in this case is designed to support US accreditation and validation requirements. In this case also we have a project manager that is dedicated to the validation, includes the workflow training, guidance and optimization, template documentation, samples and controls if the customer has decided to go with this extra service, data analysis, and at the end, customer receives a validation summary template. If we go more in detail in what is included or what a customer can expect from an analytical validation regional, customer will be running up to 8 controls, up to 64 samples in case of somatic, hematological, germline or custom assays, or up to 32 samples in case of liquid biopsy panel. Includes the project management, which will be involving the design of the analytical validation plan. If requested, we can also offer an SOP and a standard operating procedure. We will offer to the customer the chip schedule and the dilutions weekly, and we will review them in the weekly calls to assess the progress in the lab. We will do the data analysis, and we will do at the end the final analytical validation report and offer the customer the full set of data. What can a customer expect from an analytical validation report? Performance parameters that are evaluated during analytical validation are summarized in this slide. We are going to assess the performance of the assay and the system at the customer side. We will do that using the sequence level metrics, DNA library, and RNA library metrics. We are assessing the ability of the assay to call accurately variants with the assay's target range or specificity. We are determining the error rate of targeted variants that are covered by the assay, SNVs, copy number fusions within the lab, or variant calling accuracy. We are assessing the limit of detection or analytical sensitivity. We will assess the variant calling accuracy for the same sample within a single run or repeatability. We are finally assessing the variant calling accuracy of the assay across different runs and run by different operators or reproducibility. In summary, the NGS Analytical Validation Research Consulting Services is helping customers to accelerate the launch of their NGS assay in their lab, is helping customers to reduce cost, and facilitates the documentation that the lab needs. If you need to know more about our services, please contact us at Thermo Fisher Scientific. Thank you very much.
Thank you, speakers, for your informative presentations. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have a question you'd like to ask, please do so now. So let's get started. Now, we have a lot of great questions coming in. Luciana, I'd like to start with you. Now, you touched on the different methods available to study DNA methylation, genome-wide versus targeted. Why did you decide to go with a targeted approach in your studies?
Thank you for the question. And as for many laboratories, the choice of a technical approach is linked to the current research topic, the resolution required for the study, the quality and quantity of sample, and finally to the technology available in-house. The amount of DNA obtained from small FFPE tissue in our cancer research is generally low and shows a high level of degradation. So it does not allow to use a genome-wide approach for the study of methylation. So we decided to use the targeted NGS approach, since this high-throughput technology could provide epigenetic information by analyzing simultaneously multiple CPG sites of many gene regions of interest, starting from low quantity and quality of biological material.
Very good. Thank you, Luciana. And Cristina, I'd like to come to you for this next question. How long do analytical validation services take? And how much do they cost?
Well, we thank you for the question. First of all, the speed of an analytical validation services that we are offering is always depending on the customer, really. We are going to have the speed that the customer is giving us. If a customer is committed for the project and have the possibility to run between 1 to 2 runs per week, we will be able to complete the analytical validation in around 6 to 8 weeks. Of course, there are customers that are really in a hurry, and they are asking us to have even shorter times. So if they can run even more than that during the week, we are also able to complete the validation in 4 or 6 weeks. Depends completely on the possibilities that the customer has, and we will adapt to that. That we can offer the new plan and customers can start running again. In terms of the price, it's very obviously it will depend on the assay, depends on the package that the customer has selected, depends on the region. And my recommendation here will be that customers are going to thermofisher.com /analytical validation or AB, and they have there the possibility to select or ask for a quote that will be offered by one of our analytical validation specialists.
Thank you, Cristina. Luciana, let's jump back to you. Will you please elaborate on how you selected the CPG sites to include in your panel and the panel design process?
Okay. Yes, targeted gene region were selected by reviewing our lab scientific data and published report on epigenetic and solid tumors. So we specifically included genes related to oxidative stress in cancer and gene related to immunotherapy. And this selection was then finalized with the White Glove team who supported us in the definition of panel coordinates and estimation of the panel size to limit the cost of analysis. And finally, Thermo Fisher support team helped us in the optimization of the library preparation and sequencing, and with data interpretation using the methylation analysis plugin that is free available on the Torrent Suite software.
Thank you. And Luciana, let's have you take this question as well. How much
As mentioned, the methylation analysis plugin is available for free on Torrent Suite software, and it is very user-friendly. So you do not need extensive bioinformatics training. Of course, having some exposure to bioinformatics will probably help in any case when dealing with NGS data. The use of methylation analysis plugin require, I think, more or less the same bioinformatics experience as for manipulating data from single nucleotide variation or gene fusion analysis by NGS using the Ion Torrent suite. Importantly, the methylation analysis plugin allows a friendly interpretation of DNA methylation results, especially for those groups not having a strong epigenetic knowledge.
Thank you. Cristina, let's have you answer this one. What happens after the AV service is complete? Do you check in with the customers afterwards to review how the assays are working?
Well, that's a good question and a very relevant one, definitely. Customers know already that Thermo Fisher offer a comprehensive service. Sorry. So that's meaning that we are never left going to leave customers unattended after a service is finished. We have our support colleagues that are the field application specialists, the clinical application consultants, that they are going to be supporting customers after the analytical validation is done and beyond that. So customers can be reassured that they are not going to be left unattended, definitely.
Thank you very much, Cristina. Now, Luciana, you mentioned the interesting observation. Based data generated with your AmpliSeq methylation panel of non-symmetrical DNA methylation in some of your targeted regions. At approximately how many positions was this observed in your assay? And what do you think that means?
This is a very intriguing question. At this moment, I can say that the asymmetrical distribution reported and observed in our sample is not homogeneous and could be different among gene, region, and among samples. I think this is a very interesting data that could be related to the neoplastic process.
Interesting. And can non-symmetrical DNA methylation patterns become biomarkers?
Maybe. I think that anyway, more investigation on large cohorts are demanding to a better definition of this observation.
Excellent. Thank you. Cristina, what steps in the process do researchers usually experience the most time savings when using your AV services versus doing everything themselves?
That's something that a lot of customers actually are asking. I think in the presentation, we had a slide where we were mentioning about this part. I think during the pre-validation preparation part, we are obviously offering the analytical validation design of the plan. We are already selecting the controls for the customer so that they don't need to do this part that's taking time and money to do this selection and testing of the controls that are going to be running. So I would say that in the pre-validation part, this would be the saving time. During the real validation, we are obviously, I think, the main part will be the saving in the analysis, because obviously we are doing the analysis for the customer every time that there's a run, and we are preparing the every schedule for every run and the dilution, so that the customer only need to do the pipetting really in the lab, and at the very end, obviously, we will do the final report and prepare the data set for the customer in a way that it's easy to use and to obviously for the following up that they are doing with accreditation or whatever is the next part that they do with the data. Yeah.
Thank you, Cristina. And Luciana, let's wrap this with one more question. Now that you have designed and tested the custom DNA methylation panel and are starting to see the results, what are the next steps you and your group are taking?
Okay, thank you for the question. Further improvements are certainly planned for this panel, such as extending the number of genes relevant for clinical research, such as those genes whose methylation has been shown to have an important role in the tumors. Moreover, we are planning additional orthogonal evaluation of the epigenetic data obtained by our NGS panel using more conventional approaches such as pyrosequencing. And finally, taking into account the size of panel and limited number of samples that can currently be analyzed in a single run, we would certainly be interested in testing our methylation panel on the GeneXus system if it will be made available in the future, with the aim of overcoming some of the current limitations.
Thank you. And thank you to our speakers, Cristina, Luciana, Giorgio, for your time today and for your important research. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for sponsoring today's educational webcast. Before we go, I do want to thank the audience for joining us today and for their interesting questions. If we did not get to your question today, we will share them with the speakers so they can try to answer them via email using the email address you provided at the time of registration. Today's webcast can be viewed on demand shortly. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you so much for joining us. Until next time, goodbye, everyone.